So, hello everyone. We are once again in the what we are reading a YouTube channel of the South American Mycorrhizal Research Network. This time around, we have something a little bit different. The paper that we are talking about today is not strictly mycorrhizal, but nevertheless is very proper to our field. Um, this article is called Global Epistasis and the Emergence of Function in Microbial Consortia. And today we have with us the lead author, the first author of this of this paper, Juan Díaz-Colunga. He's a postdoc at the Instituto de Biología Funcional y Genómica in Spain, which is also with the CSIC, with the main research institution on Spain. So thank you, Juan, for being with us. Let me start by asking the question that I ask everyone in this channel, and is how this study came about. Is your postdoc or is the bigger project? How this came about?
So, well, first of all, thank you for having me. I'm very happy to do this interview. The question of how this came to be is maybe a bit broad, I guess. The short answer would be, well, there's this idea in microbial ecology that we can treat, I don't know, let me start by the beginning. There's this idea that we can treat cells as bags of genes, right? This idea comes from genetics, and it's becoming a classical one, I guess. And so in ecology, it's starting to be talked about the notion that we can treat communities as bags of genes, right? So in a sense, there's this connection between the way in which genetic interactions have been treated traditionally and the way in which sort of ecological interactions within the context of a community have been treated. And of course, that idea, it's not ours necessarily. There are a lot of people that are proposing kind of things along these lines. I don't know, I guess the work of Sepe Kuhn comes to mind, or Karnagoda, or maybe Will Ludington. Like there are a lot of people who have proposed, sort of frameworks to think about genetic interactions and ecological interactions sort of under the same set of rules, right? And this paper specifically, this project specifically kind of speaks to this way of thinking or this line of thinking, right? So in a way, it's a natural sort of development of events. But yeah, to talk about more the specifics, I joined the lab I think it was 2020, in late 2020. And then we started working on this maybe in 2021, a year after I joined or something. It was just a matter of, the original idea was sort of conceived by Alvaro and by George Bajk, who was a postdoc in the lab at the time. He's now an assistant professor at TU Delft. And I guess, I don't know, they were having a coffee or something, and they started talking about this kind of idea. And they thought, hey, maybe this global epistasis thing could be applied in ecology, right? And then I joined and I started doing some analysis, and we saw that it worked. And so we got a few more people involved in the project. And yeah, that's kind of it all started.
So you just mentioned a central concept in the article, which is epistasis. And yes, originally, like when the people from the modern synthesis started to look at like Mendelian inheritance, in particular, Sewell Wright noticed that things may be more complicated and that the genes interact a lot. I recently saw a paper in Evolution showing that they reanalyzed like genomic data and yeah, they found that epistasis is very common, is everywhere. And a main point in your article is that there is this analogy between genetic epistasis and ecological epistasis. Can you explain us a little bit more in deep what you mean by this?
Right. So, the thing is, I'm a physicist. Alvaro is a physicist as well. And, in the lab, we've had people who have very different backgrounds, right? We have, of course, microbiologists, but also, mathematicians, computer scientists, etc., right? So from our perspective, it's very common within physics to think about complex systems from the perspective of complex systems, right? Like we can sort of forget about the details and stop looking at whether the parts that are interacting are genes within a genome or species within a community, and just if all we see are things that are interacting, and then we kind of forget about the underlying mechanism that may be governing these interactions, then we can maybe start using frameworks that are not commonplace in some fields, but for sure, could be commonplace from a complex systems perspective, right? And so in a way, it's very natural. Like, of course, if we think about the biology of it, there's no reason to believe that the sort of mechanisms, the molecular mechanisms governing genetic interactions are going to have anything to do with the mechanism governing ecological interactions, right? Like, of course, there's going to be some crosstalk there, but in principle, a cell and a community are different things, right? So biologically, there's no prior for us to think that these things should be studied under the same framework, right? But then if you kind of take a step back and just look at things, elements that are interacting, and you sort of define these interactions in a more sort of mechanism agnostic way, right? Like we forget about whether the interaction is due to maybe competition for a resource, or maybe it's due to like the physical proximity between two residues of a protein or something. If you forget about those details and just look at emergent effects, like, oh, I have mutation A and mutation B, and then I combine them together, what's their effect, right? I could ask the exact same question in the context of ecology, like I have species A, species B, what happens if I put them both together in a community, right? And so it's at this point when we kind of take a step back and maybe forget about the details, or maybe not forget is perhaps the wrong word, but like if we don't necessarily aim to understand every specific detail about the biology that's going on under the sheets, then at that stage, maybe we can start applying common frameworks to both problems, right? And perhaps that's the main take-home message of the paper, I guess, which is if we want to start understanding something, it's maybe not the best idea to start by the sort of lowest units of organization. Maybe we can kind of zoom out and look at emergent properties, and we'll get somewhere. We'll get farther away with that kind of approach.
Yeah, no, I totally agree. I was thinking that usually the way that most people thinks about causation in these frameworks is from the bottom up. But what you are saying is that emergence of like this case of global epistasis, the causation should be studied at the higher levels, right?
Yeah, it's something. I mean, it's along those lines, and I'm not saying like, of course, we're scientists and we want to understand how things work, right? And ultimately, it is. I'm not saying there's no value in trying to identify the specific mechanisms that govern these, be it genetic interactions or ecological interactions. Of course, there's value in that, right? But maybe that approach can be complemented by a different type of sort of strategy, which is looking at maybe properties that are more emergent and patterns that can be interpreted maybe or in a statistical sense rather than a mechanistic sense, right? And that's kind of what we are trying to do.
And in that sense, what is the difference between like epistasis as one usually have it in mind, and what you call a global epistasis?
Right. So as you were saying before, right, it is definitely true that genetic elements interact with one another, and mutations and genes and signaling pathways and metabolic pathways, and that is all very complex, right? Like a cell is very complex, and so the question is, well, I mean, I guess it's a reasonable expectation to have that since there is all this complexity, well, yeah, maybe we know a lot about this particular gene or about this particular protein or in general about this specific cellular element, right? But that does not necessarily bring us any closer to understanding sort of emergent phenomena of the cell, like life itself, right? because there is epistasis, there are interactions between elements. And if we kind of take the long road and start trying to characterize these interactions, then, we have to take every pair of elements and then every trio of elements and then every quartet of elements and so on. And of course, you have such a complex, such a huge system that is not very, it doesn't sound very reasonable at first, right? And so the idea is, well, what can we do about that, right? Like, how can we make predictions if there's such underlying complexity and it's so difficult to untangle it, right? The idea is that the complexity of the interactions do exist, but they are not necessarily random. They often have some underlying structure, which means that there exists this sort of emergent phenomena that we can characterize without necessarily looking into the details, right? And that is kind of what, I guess, hides behind this concept of global epistasis. The idea is that, sure, there is epistasis, which means that, pairs of genetic elements can interact, trios of genetic elements can interact. But there is this global component to epistasis, which means that at the end of the day, even if I don't fully know how that interaction landscape looks like, there are emergent patterns that I can characterize, right? Like that there is, and in the case of global epistasis in particular, what we see is that the fitness effect of a mutation is often predictable from the fitness of the genetic background where that mutation is included. So in a way, it's like I do not need to know all the molecular level details about how that mutation may or may not be interacting with other genetic elements of the genetic background. I just need to characterize this sort of emergent pattern, right? And with that, I'm able to make predictions to some extent. Of course, there's going to be variability and whatnot, but I can make predictions, even forgetting, or not even, but when I forget about the details, I begin to be able to make predictions, right? And so that's kind of why the global word is put there, I guess.
So before going into the questions about the two kind of big experiments or one experiment and one data analysis that you did, you just mentioned something very, very interesting regarding the genetic background. And I was wondering if this implies that to at least with your data and within this framework to predict the function of any community or well any microbial community, um, you just need the the presence data of each member of the community and not necessarily the abundance. What did you found regarding this?
Right. So the thing is, I mean, that has to do with the specific way we are conducting our experiments and the specific data set that we're looking at, right? The data that we are dealing with kind of mapped the composition of an inoculum to the function of the community that assembles from that inoculum without necessarily, I mean, when I say community, it's maybe a wrong choice of words, because in our experiment, for instance, we only let the microbes grow for 24 hours. No, I think it's 48 hours, actually. So the community is not in equilibrium. And there's definitely an expectation that if we had kept propagating these communities, then, there will be some ecological dynamics making it so, maybe some microbes would have gone extinct or some others would have increased in abundance or whatever, right? So in a sense, maybe the word communities, maybe not the best choice of words, just because there is no community in the sense that there's an ecological stability, right? Like that is not the case in the experiments that we are conducting and in the data sets that we were looking at, right? In fact, one of the things that we are aiming to do now is trying to apply the same set of principles to communities that are sort of stable in time in terms of their composition, right? But in this case, the reason why we are mapping presence absence and not necessarily abundance is because we have full control over what we inoculate within our system. So we can take, I don't know, a set of 5, 6, 7, 12 species or whatever, and we can inoculate them and then let that do whatever it needs to do, right? But we're not looking at the dynamics, we're not looking at stability, we're not looking at which species are going to extinct or not. We're just sort of inoculating something and then harvesting something after a certain amount of time. But in that sense, that the idea is that we do have full control over that, right? Like even if we cannot, of course, once the species are there and they're still growing and interacting, then like it's a mess, right? Like we cannot externally act on that, I mean, we could, I guess, there are ways in which you could externally act on that, but in general, the easiest thing for us to control as experimentalists is what we inoculate, right? Like what we put in there. And this is not, I mean, I don't see this as a limitation in the sense that there are a lot of biotechnological applications that do work like this. You know, I have a bioreactor, I'm trying to ferment something or whatever. What I can control is what I inoculate into the system, right? Whatever it does after that, well, then that's kind of not my problem in a way, right? But the sole idea that we can sort of predict what's going to happen when I include a new species within an inoculum, I think that is powerful in itself because that allows me to sort of make predictions beforehand. Like I don't need to see the dynamics of the community in order to be able to predict what the function that I'm going to harvest is going to be, right? All I need to do is sort of control the time zero of the experiment.
That is indeed very, very powerful. And it leads me to ask you specifically about the experimental part of your study, about the kind of taxa you use, the kind of medium, and also the type of functions that you measure. Because, yeah, I immediately ask myself if this is applicable to vastly different microbes or vastly different mediums, or as you just said, if the measurements are taken not 24 hours but weeks or months later. So can you and also after that maybe mention the uh, you did already, but maybe reiterate the main finding of this part of the study.
Right. So in terms of our experiment, I mean, there's nothing really particularly interesting about the species that we chose. I mean, there's some history of the lab, like they came from a previous experiment. And in that experiment, there was a reasoning as to where we sampled from and the type of medium that we used. But at that stage, like when we conducted this experiment, we were very much at a sort of proof of concept stage, right? Like we just wanted to see if it worked. So we kind of took 8 species, like 8 isolates that we had hanging around in the lab. And of course, you know, there is some, there was some prior filtering, you know, like we wanted to make sure that these bugs were able to grow in the lab conditions that we were growing them and so on. But there's nothing really particularly interesting about them, you know, like, and in a way that is, we wanted to be honest with ourselves, right? Like it would have been easy to, I mean, not easy, but it would have been doable, I guess, to kind of select specific microbes that were going to behave in a certain way so that we would see certain patterns and whatnot. But in this case, it's like we literally just took whatever we found and we used the medium that we had been using for a bunch of other past projects, and we just saw what we got. What we did want, however, is that the function that we were looking at was truly complex in the sense that it was truly sensitive to interactions, right? And that is why I guess your other question as to regarding our choice of function. Well, we measured the amount of pyovertins that the communities were secreting. And the reason for that is that, well, first of all, it's very easy to measure. Like it's just that the pyovertins are kind of this, I guess, yellowish, greenish color. And so it's just by, you know, measuring optical density, you can get a pretty good estimation of the amount of pyovertins that are in the spent medium of a microbial culture. So that is one reason, an important one, I guess. But then pyovertin secretion is actually very tightly regulated by quorum sensing. Of course, there can be, you know, effects on population size, meaning that even if a species secretes the same amount of pyovertin per capita, of course, once you start putting that species in the context of different communities, the population size can change. So the total secretion, the total contribution of that species can also change. And, you know, pyovertin secretion is actually a interesting on itself, like like this type of molecules that they take part in, you know, iron uptake, and you know, recently there are people saying that, or like proving that they have to do with cross protection against antimicrobial agents and whatnot. So it's an interesting function, but from our perspective, what we cared about was first of all that it was easy to measure, and second of all that it was truly sensitive to interactions at the ecological scale, right? And so in that sense, this one kind of fit the bill perfectly. So that's why we used it. It was after that we asked, well, you know, but how general is this? And that's when we went to the literature to analyze other data sets.
And before that, so which was the main result in this?
Right. So what we saw is that the same patterns that predict the fitness effect of a mutation can predict the effect of a species in a consortia, right? So this idea that I was mentioning before, that I can forget about, the mechanistic details and just look at these sort of global epistasis patterns, these correlations between the fitness effect of a mutation and the fitness of the genetic background. So the same sort of correlations emerge when you look at the effect, what we call the functional effect, meaning the change in function at the community level when you sort of co-inoculate a new species. So in a way, acquiring a mutation for an organism is analogous to sort of including a species for a community, right? That's sort of
that's a very nice way to like summarize the main message of this study. And did you find the same when reanalyzing published data, and which kind of published data did you reanalyze?
Right. So the first thing to notice is that there are not that many data sets of this form, right? And in fact, one of the things we're dealing with now in the lab is sort of developing sort of experimental methodology in order to be able to generate this type of data in high throughput, really, right? And it is true, I've done a bunch of these experiments when you have to like sort of mix different combinations of species and measure something and whatnot. And it is very sort of labor intensive, even with relatively small sets of species. It's really hard. I mean, if you think about it, like you cannot escape combinatorics, right? And so if you have 20 species, then you have 2 to the 20 combinations, and that's a lot. And there's no way around that, right? And so, sure, there are alternatives, like people have been developing sort of microfluidic devices that allow you to actually test, a wide range of different species combinations and whatnot. But in general, it's difficult to do, right? And so there are not that many data sets of this form in the literature. In the paper, we did sort of reanalyze whichever we could find. We did find a few more as preprints, which we did not include in the paper, just because, we didn't want to use anyone else's data before themselves. But yeah, the bottom line is there are not that many data sets of this form. And one of the things we're concerned now is sort of trying to increase the number of data sets of this sort of with this structure that are available. Because I do think that generating that type of data is important, or is sort of a bottleneck that we're facing now, not just from our perspective or from the global epistasis perspective, but for sort of microbial ecologists in general. If you want to disentangle the complexity of microbial interactions, then you do need this type of data. And yeah, we do need to make an effort as a community to sort of step up our game in this sense.
But overall, these few studies that you found lead to the same conclusion as your experimental setup.
Right. Yeah. We did find these patterns like sort of all over the board, right? And what is interesting is that we did find a bunch of different behaviors, meaning that these patterns that I was describing that allow you to predict functional effect of a species and whatnot. We did see that they were very dependent on which species you were looking at, which set of species you were assembling your combinations from. Of course, the environmental conditions under which you were growing your communities, the specific function that you're looking at, and so on, right? But in general, we did find that these patterns emerged quite ubiquitously. And so that was kind of inspirations for us to think that, well, maybe our experiment was not a fluke. It was actually something that we may expect to happen sort of generically, right? Of course, this is still an open question, right? Like I was saying, there are not that many data sets, which means that for sure, maybe we could think about other types of functions or other libraries of microbes or other environmental conditions under which maybe this will not work. So there is a question as to when can we break this, right? Like how can we design experiments so that we don't see this type of thing, or we don't really see that the effect of a species is predictable, right? And that remains an open question.
Oh, interesting. Yeah, that last point is very interesting because actually in the second half of the results and in the discussion you go into how universal this mechanism can not mechanism but this pattern is, and in general you talk about the emergence and the ubiquity of this global epistasis. So can you explain us a little bit about how specifically, because this is, yeah, of course, this is an emergent property. So how is originated?
Right. I mean, so we know, it's not that we know, right? Like other people, particularly from the field of genetics, right? Like people like Desai, have been working on this type of problem, and they have been able to make sense of where these emergent patterns come from in terms of specific interactions, right? Like it's not sort of two opposing hypotheses, like either things are global or they are local in a way. It's kind of both, right? Like local interactions in the sense that pairwise, third order, fourth order, and so on interactions do exist. The thing is, when they sort of act together, they give rise to these emergent patterns. And that connection has been made in the field of genetics. And in fact, in our paper, we show that the same logic, the same math, so to speak, can be applied to ecological interactions as well. Again, once you forget about the details or the specific elements that are interacting, all you're left with is interacting elements, right? So you can forget about whether an element is a mutation or a gene or a species or even a bug of species or something like that. If all you're looking at are interacting sets of elements, then the same sort of mathematical framework can accommodate many sort of layers of organization, right? And so in our paper, we kind of adopt the same approach, the same framework that these people from genetics have been using to kind of make that connection between fine-grained interactions and the emergence of these global patterns, right?
Mm, Great. I have a couple of last questions, and one obvious one is the applicability of this framework. Because as you said earlier, and especially in my field of mycorrhizal ecology, yeah, there are dozens and dozens of products already. Sadly, most of them are not well regulated, and many of them simply do not work. So, do you see an immediate or mid-term application of this framework in any type of inoculants, but in particular like soil microbial inoculants?
Right. I mean, it's a very good question and definitely one of the directions that we want to move towards. I don't know, I could be talking about this for, I guess, hours. The short question is, well, the answer is going to be determined by the extent to which there exists a mapping between what I can control, which is the inoculum, and what I want to harvest, which is the final function, right? So for instance, if I inoculate something and then want to harvest a function, I don't know, 2 years later, and then that function is sensitive to temperature fluctuations or humidity fluctuations or whatever, things that I cannot control, then maybe it's not so obvious the existence of that mapping, right? Like I can inoculate a same set of species twice, but then harvest two very different functions just because there are another million variables that escape my control, right? So in those cases, I think, we need to rethink our approach to these type of problems. The good news is that in biotechnology, this is not always the case. Like there generally exist cases. I don't know. I guess I always give the example of like food or wine fermentation, right? Like where what happens is literally you inoculate something, you wait for whatever amount of time, and then you harvest your product, and that's it, right? Like you have sort of a greater deal of control over the parameters that could sort of disrupt this mapping between composition and function. But I guess the short answer is whenever there is an application where that mapping between community composition and function exists, that is kind of a battle that we're willing to fight, and then, well, for sure, as I was saying before, maybe we'll find some functions where this breaks, the function is very complex, or maybe the set of species that one needs to work with is huge, so we would need to sort of rethink strategies. But in general, I would say that the main sort of requirement is the existence of this mapping between composition and function.
I see. And as you said, yeah, this can be very complex. I imagine, I don't know, having 20 fungal species and making all the combinatory approaches could be huge. In my view, what I have in mind is that at least in our field, this has not been done like thoroughly and with a really nice experiment as you show. Um, well, the final question is to know what are you doing right now? What are the next steps in this study or in this field or in other fields that you are investigating?
Right. I mean, I guess we've talked a little bit about it. There are like many directions that we are aiming for at this point. Um, I guess more on the theory side, there are a bunch of open questions that we need to address regarding, well, what requirements or sort of what conditions will yield the emergence of these patterns? When can we break them? What interaction structures do we need to look at in order to, get one patterns or another or no patterns at all? So that's more on the theory side. Then there's also the issue with what we were talking about before about ecological equilibrium, which is, well, sure, we've shown that this works sort of in closed bioreactors over a predefined period of time. But what about, say, natural communities that are stable in some sense, at least, right? Will it work there? Is it the case that if I throw in a new species to one of such communities, is that species going to have a predictable effect or maybe not? Or is it going to depend on some other variables that we're not looking at? So that's maybe more on the theory side. On the experimental side, like I mentioned before, we're working on trying to develop protocols to be able to significantly expand the number of data sets that we have available to play around. And then, sure, as you were saying, like we're definitely open to trying to tackle some real open problems out there, right? Like the functions we've been looking at so far, maybe more, not so interesting in from a biotechnological perspective, I guess, just because we were starting this and at the proof of concept stage, we were aiming for functions that were easy to measure and whatnot. But I do think that there's definitely interest from our lab to apply this to specific problems, like real world problems, so to speak, right? So yeah, that's kind of the two kind of branches where we are moving right now.
Maybe in one last thing, maybe in the theoretical side, and because it's my bias and because I'm working on these issues over the last year, uh, have have you thought about the evolutionary forces or consequences of this framework?
Right. I mean, again, there's a lot to talk about here. Evolution is one of the things that we don't know how it may or may not affect this, right? Like I was saying before, suppose that I inoculate a set same of species, and then I wait 2 years and I harvest my function, right? And in between, God knows what happened. Then maybe this mapping does not exist because whatever I inoculated and whatever function I harvested has nothing to do with one another. Evolution can do the same sort of thing, or maybe not, right? Like I can inoculate the same set of species, but then if I give them enough time to evolve, then the function that I'm going to harvest is going to depend on how sort of reproducible those evolutionary dynamics are going to be, right? If I take two communities or two inocula that are exactly the same, exactly identical, but then evolution is going to sort of make them diverge and make it so the communities that end up sort of assembling there are going to follow totally different evolutionary paths.
Yeah.
Then maybe the function that I'm going to harvest after whatever amount of time is going to be different as well, right? So evolution may disrupt this, or maybe
Even
not. Maybe evolution will happen, but it will be sufficiently sort of conserved or sufficiently predictable so that the function that I will harvest, even if there has been evolution in between, will be the same. And again, this is an open question, right? So this is only one of the sort of points of intersection between sort of evolutionary theory and maybe questions that are open in the field of evolution and what we are doing, right? But there are many others, and again, we would need to be talking for 2 more hours to unveil them all.
these two scenarios that you just mentioned remind me to Stephen Jay Gould and the possible worlds, because yeah, I agree that it can be the same, can be different, even if the conditions are exactly the same and the initial composition is the same, it can have two very different evolutionary paths.
Right.
Okay. Yeah, as you say, we can talk about this for hours, but I think that I'm closing in the interview now. thank you so much for your time and for introducing us to the mycorrhizal community to this very interesting approach. And thank you.
Thank you for having me.